3 A-Level Biology 2018. So, starting with question 1, we've got broken bones are replied by cells called osteoclasts and osteoblasts. And we've got information here about the cells that it's followed up by the figure 1. So we've got the information that those cells secrete hormone called osteocalcin, and it's in the inactive form. Okay, so where can we see this exactly on the diagram? We started from here. They secrete the hormone which is in the inactive form. Okay, so it's a protein, and the active form of this will bind to the receptors on the beta cells. But how can we get to the active form? We need to change the pH. Okay, so change in the pH is going to make this hormone active. So remember, with the acidic pH, it changes, for example, the tertiary structure, so it could affect the hydrogen bonds, and it's going to make it active. What's the job of the B cells then? They will secrete insulin. Okay, so that's the explanation of what is taking place here. And we've got the figure 1 that shows you the production that it's controlled by positive feedback. What do you need to remember about positive feedback? Idea of more. Okay, so more things will be secreted or released. So let's have a look at the questions now. So you've got question number 1, that the secretion of the hormone in the inactive form, okay, by osteoblast, it's controlled by positive feedback. So what did we say? We need to think about more. How do we get to the active form? Of course, by changing this pH to the acidic. So using the information from figure 1 that we just were looking at, we need to explain why this is a positive feedback. So in other words, use the figure 1, use the definition of positive feedback, and say what you can use from this diagram to explain the positive feedback. So what we could see, and what we've seen a few seconds ago, is that the osteocalcin causes more release of insulin, so this approach of more for positive feedback. So more insulin causes more inactive osteocalcin release. So can we see this? Right? More insulin stimulates production of more inactive form. Lovely. So that's done. So we're looking at the next question. So the acidic pH conditions created by osteoclasts cause the inactive form of the protein, okay, to change into the active form. So what we need to suggest, give the reason how is it possible. So with the questions suggest, you're looking at the statement that they've given you, and then you're using your biological knowledge to think, right, what could possibly happen for this, okay, to take place. So proteins, we're thinking about the structure. What's the reason of becoming from inactive to active? Here we're thinking about the induced fit, for example. What happens? So they change in the pH, could break the hydrogen bonds, so that changes the tertiary structure of that protein, because it was information that this is a protein. Right. 1.3, then binding of insulin leads to an increase in the rate of respiration in cells such as osteoblasts, and explain how. So increase in respiration. What do we need for what do we need for respiration? We need glucose. We know it was the positive feedback that we were looking for. So increase means we need more glucose. Okay, how the glucose is then related in the whole thing? We've seen insulin. What is insulin? Okay, insulin leads to more transport proteins for glucose, for example. So more glucose will be available for respiration for those cells, so they enter the cell. Right, question number 2. Question number 2 is another long story to read. Okay, so we've got CLL is a cancer that affects some B cells, okay, and a drug, it's used to treat the CLL. It binds to a protein called CD20 on the surface of B cells, and if enough of that drug will bind to a B cell, they will kill the cells. But what's the problem with the drug? Is the fact that they can kill both healthy and cancerous B cells. So the body then will produce new B cells. So the amount of CD20 protein on the surface of B cell varies. That means it's different, and that's a really tricky thing that examiners like to put. They like to use the words like different, only one, okay? So make sure you pay attention to those words, might be important. So doctors investigated the relationship. What's the relationship? Okay, it's obviously we will be properly looking at the correlation between the amount of the CD20 and the B cells of a patient and the percentage of B cells destroyed by the drug. And the results are shown here in the figure 2. So firstly, how to approach the figures before you look at the questions. So look at every single axis, what do they tell you? And then think about the pattern. So what is the pattern here? It's of course a relationship, as they told you. It's a correlation. What kind of correlation? Positive. Because once one increases, other one increases as well. So then it's a question, what statistical test could be used? Okay, and you need to give the reason of your answer, and they even use the word relationship, so kind of identify what is the test, by the way. So name of the test, it's correlation coefficient. It's only so we can choose between chi-square test, t-test, or correlation. Of course, if you're talking about relationship, it's a correlation, and that's the reason, because you're looking for the relationship between the variables. So that's a quite nice question. Next, we've got from the data, what can you conclude? And this is a statement about the effectiveness of the drug. Okay, so what can you conclude from the data? You only are allowed to say what you can see. So conclude any type of the experiment, any type of investigation, you just are allowed to use the information and say literally what you can see. That's how easy it is. So they've told you that they are not interested in any statistical analysis. It's just what you can see. So from that information was obviously text and was the graph, what can you see? So what we can see, okay, it's more CD20 on B cells, then it's the higher the percentage of destroyed B cells. Correct, we see that from the graph. At the best, only destroys about 80% of B cells. Correct, you can see that on the graph. So we don't know what is the total percentage of the cancer cells within the experiment. We don't know, because what they told you was the fact that they will kill both good and cancerous cells. So we again we don't know if that will care for cancer or not, because what we could predict within the maximum 80%, let's say the other 20 that weren't killed, could be cancerous cells. So little effect below about 5 proteins on the cell. Where can we see this? Obviously from here. So 5, it's around there. So the effect is quite small. And the maximum, okay, what we've said, it's 80. Yes, it is. Okay, so let's have a look at the next question. So the next question tells you that approximately 11% of people with CLL also have mutations of a gene called not CH1, okay, and this leads to the non-functional transcription factor. So if it's a non-functional transcription factor, that means that transcription is not taking place, so you do not produce the proteins, okay, and the results, okay, with the mutation and without the mutations were shown in the table number 1. So in the table number 1, we've got the percentage. So be careful, it's a percentage. So what's 4? Not 0.04. What's 22? Not 0.22. Okay, so careful with what you're reading. And I told you that they contain approximately 1 × 10 to the power 9 B cells per decimeters cube. And your task is to calculate the difference between those numbers with mutation and without the mutation. So look, between the number, percentage, so changing that to the numbers. So let me open the working out for this question. So what we've got here, okay, one sec, right, so 22, okay, times 10 to power 9, because that's what they told you, that's the volume, minus, because we're looking at the difference, 0.04, so converted that percentage into the number, that gives you 0.18 times 10 to power 9. They wanted the answer in the standard form, so we remember numbers in the standard form between 1 to 9, so 1.8 times 10 to power 8. Okay, and that's the working out for this question. Right, so let's keep going. So we've got to use all the information. So you need to look through the table, through the text, to suggest. So again, give the reason, biological knowledge, okay? How, and again, that's a statement. Mutation led to difference in the percentage of B cells destroyed by the drug. Difference, okay? So what we're looking at, we're thinking about here the structure. So what mutation causing, and that's just your recalling stuff, mutation causes changes in the tertiary structure of transcription factor. So transcription factor is not complementary to CD20 DNA, so mRNA is not produced, so obviously then polypeptide is not produced, so not enough proteins, so little for drugs to bind to, okay, so B cells won't be destroyed. So that's one of the suggestions to achieve 3 marks. Right, so what we're looking at next is question 3. Okay, question 3 was about meiosis. So the woman with the first division of meiosis produced one daughter cell that has almost all of the cytoplasm, and the other daughter cells consist of a nucleus surrounded by a very little amount of the cytoplasm and the cell surface membrane. So this very small daughter cell, it's called a polar body, and polar bodies do not usually divide. That's important information, so they won't divide. And the same process occurs in the second division, resulting in one egg cell and 2 polar bodies. And they illustrated that in the diagram, and they told you that it also shows what happens in one pair of homologous chromosomes. So we're looking at this figure. Okay, and what we've got here. So this is the first division. Okay, so as the question says, we've got a daughter cell and we've got a polar body. Second division, we've got 1 egg and 2 polar bodies. Right. So what the question is to complete figure 3, putting the alleles in the box. So what they've given us here, they've given the alleles of the first polar body. Okay, so this was the pair of the chromosomes that we started with. So what are the gametes that they can produce? Okay, so this chromosome produces the dominant allele A, that one recessive allele A. So all of the offspring that we will get after this division will be heterozygous. Okay, so that's what we got it from. Right. Next, second division, what we need to remember when we're looking at the alleles, okay, it's then the fact what we've got here. So we've got one allele A, okay, dominant, which comes from here. We've got the heterozygous, which comes from there, because we know that this polar body, it's not going to divide, so of course it's going to have the same content. So that one comes from the division, and we can see that has a dominant allele, so there is no other choice to get a recessive allele in the egg cell. Right. So we're looking at the tick box here. So we need to think, what are those processes? Okay, produces the combination of the alleles. So of course it's a crossing over, because it's an exchange of the genetic material, exchange of the parts of the chromatids that leads to the combination of the alleles. So a tick box here. Then we've got the question, a scientist measured the diameter of the polar body and the diameter of the nucleus, and the diameter was this, and the diameter of the nucleus was that. And the density of the mitochondria in the cytoplasm of the polar baby was 0.08 mitochondria. And they're asking you to calculate the number of mitochondria in the polar body, and you need to assume that they are they are sphere. So they're good because they've given you here the equation, how to work out the volume of the sphere. They don't have to, because that's one of the equations that you need to know. So calculation follows as. My advice is always write down everything what they told you. So as you can see here, I wrote down everything what I know about polar body, sorry, so should be body, and diameter. And from the diameter, I work out the radius, so just divided this by 2. Same here. Okay, and then I use this equation that they've given me for volume of the sphere to work out the volume of the polar body and the volume of the nucleus. So I've got those two answers. What they were looking for, they were looking at the differences. Okay, so because they want the number, the density of the mitochondria in the polar body. So what do I need to do? I need to take away from the polar body the volume of the nucleus. So then I know that the rest will be just available for the mitochondria. So that is 409.17. Then I've got the volume available for the mitochondria. Multiply this by the density of it, which is 0.08. And the number of the mitochondria I've received, it's 32.7. So, going for next question, okay, we've got the disease that it's caused by faulty mitochondria. All of a person's mitochondria are inherited from the mother, okay, and they're giving you idea that 3 times 10 to power 5 mitochondria will be in that X cell. So treatment to prevent this passing, okay, to the next generations will be the removing of the nucleus from an egg donated by a woman with healthy mitochondria, or replacing this nucleus with the contents of the polar body from a woman whose egg cells are affected by disease. So suggest again, give the reason. Okay, give the reason. Look at the statement. How this treatment prevents, so other words, stops inheritance of mitochondrial disease. Okay, so reason why. So for example, the egg created has a nucleus and DNA of affected woman, so it could be one of those, and then it has many normal mitochondria of unaffected women. So that's a logical explanation of how we can prevent, so stop passing on this genetic information to the next generations. So we're looking at last part of question 3. If most of the mitochondria in a cell are faulty, this prevents, so again, stops many important enzyme catalyzed reactions taking place. So now you're thinking, how possibly, okay, now can I combine enzyme and mitochondria? What happens in the mitochondria? Respiration. What's the product of respiration? ATP. What does the ATP got to do with the enzymes? ATP provides energy for enzymes reactions. Phosphorylations taking place make the substrates more reactive. Okay, so that's the logical reason for this statement. Right. Going for question 4 now. So binding, look, another question with binding. Binding of one molecule of oxygen to hemoglobin makes it easier for the second molecule to bind, and explain why. Explain, say how. Use your biological knowledge, so thinking about structure here. So binding of the first oxygen changes the structure of the hemoglobin. So here you need to think about this cooperative binding. So changing that, why is it easier for the second to bind? Because it uncovers another binding site. How many binding sites do we have? 4. So first binds, uncovers the binding sites for the second and for the third, and then obviously the fourth, it's still hard to bind, but it does take place. So we've got here another long question with the experiment. So we've got a special microscope slide which can be used to count the number of blood cells, and they're giving you lots of information. So the surface has many of the slide has many small equal-sized squares. The depth of the liquid, that's important because you can work out the volume using the depth. It's not 0.1. And when counting cells that touch top or left lines are counted, but cells that touch right bottom are not. Right? So right bottom, them ones are not counted. So the doctors use them to determine the number of red blood cells per and the unit. That's super important. All those units identify something. Okay, so he diluted the original blood sample by a factor 200 times before putting it in. So what we've got, how can we possibly work out the volume? 0.05 here, 0.05 in there, times 0.1, because that's the depth of the blood cells. Okay, so that's something to remember. So looking at the question, is doctors counted the red blood cells in many squares. The mean number was 7, and the original sample was diluted by the factor 200. And look, they again want the answer in the standard form. So what we've got here, firstly, what I work out, it's the volume. Okay, so in here, what did we use to work out the volume? The length of the square. So we got 0.5 times 0.5. 0.1 is the depth. So that gives you the volume. So then we divide this 1 over this volume, which gives you 400. Okay, and where the rest of the numbers come in? They say that they diluted this by 200 times, so I multiplied this by 200 times then to get my original answer. And 7, where is 7 coming from? Because the mean number was 7. So in my final answer, I've got 5.6 times 10 to power 6. Okay, so that's the working out for 4.2. Right. So 4.3 then, when counting, cells that touch top or left lines are counted, but cells that touch right or bottom are not counted. And give the reasons, give the reasons for this rule. Okay, so why would you not count them? What's the reason for this? Okay, so of course you want to compare your results, and another thing, you don't want to count them twice. Okay, so that's the reason why would you not count those. Next part of this question, it's the doctor wanted to know how many white blood cells per this unit of volume there were in the different sample of blood. So again, they diluted by factor, this time 20 times, and he then made the white blood cells clearly visible by using a stain. Okay, and the nuclei appear dark blue. So when counting white blood cells, the doctor only diluted the blood sample by a factor 20 times instead of 200 times now. Why? And when counting red blood cells, what you need to remember about the difference between white blood cells and red blood cells. White blood cells, they have a nucleus, but red blood cells, they do not, okay? But also, we're looking at the numbers of those, okay? So numbers of those will be the fact that there could be possibly less white cells, okay? So there's no reason to dilute them that much, okay? So that's the reason. Explain how the stain allowed the doctor to count the white blood cells, okay? Because what we've mentioned a second ago, the white blood cells have a nucleus, but the red cells don't. So that stain will be visible for white blood cells. Right? Going for question 5. So scientists investigated the role of protein in mitosis. Okay, so they involved the cell fractionation and ultracentrifugation, and the scientists began by lysing. They told you what lysing is: breaking open cells and organelles using a detergent, and that dissolves lipids in water. So just how? Okay, it releases. So, other words, nothing else than your year 1 content. Know the stuff that you need to release the organelles. So, cell membrane, it's of course made of phospholipids. So we need to dissolve the membrane. Right. Explain how ultracentrifugation will separate those from other molecules. So we need to spin it at high speed, and will separate molecules, separate the organelles by by the mass, by the size, by the density. So that's your year one, really easy, lovely content question. Next, we've got the information about spindle fibers in mitosis, okay, and they are made of the protein called tubulin. So scientists treated cells in the culture with small interfering RNA. What does that do? You should remember that prevents translation. So you should write this down straight away. And this siRNA causes RNA interference of expression of this gene. Okay, so the scientists took samples of cells at 0, 48, and 47 hours after adding this siRNA. So then they used the gel electrophoresis to separate this. How can we separate? Remember, by size, by mass, by charge, by length. So figure 5 shows you the results of the electrophoresis. So that's what we've got. And so that's how the siRNA produced those results. Okay, so we know that siRNA prevents translation. How? Because it will bind to mRNA that codes for this gene, and that will prevent translation. Okay, and reduce. So if that's the case, they will reduce the tubulin production, because they told you that this expression is needed for the tubulin to be present. Right, question 6. Ammonia in soil is oxidized to nitrates and nitrites by species of nitrifying bacteria. Okay, what's oxidation? So it's the oxidation is loss of electrons or hydrogens, but gain of oxygen. So you should think about this. So again, we've got different pHs. And we've got the information here about the soil from site A and B, difference in the pH. And they measure the concentration in soil sample over 20 days. And the same concentration of ammonia was used at the start. And the mass was recorded. Okay, so we've got lots of things going on here. So we've got the concentration of ammonia. We've got time. We've got the soil A, okay, soil A here with the pH, B at the bottom, and then the mixture of those. Right. So what the questions are like, they're asking you why the scientists use the units as that. So micro, it's a prefix and it's quite small. So why would you use that? Because there was a little amount of ammonia. Right. And why would they use grams? Because you're comparing. So you're comparing, okay, per grams. This is why we're using that. And then we need to calculate the difference of breakdown between days 0 to 2 in soil A and soil B. So you're reading off the graph, okay, what's the difference? So coming back to the graph, starting between days 0, okay, and 2, draw the line and then read off. So both of them started at 20, but then we've got a difference. So for A, after 2 days we've got 11, so the difference is 9. For B, after 2 days we've got 2, so that's the difference 18. So took those away, so 18 minus 9, it's 9. And of course we need to provide the units, and they want it per day. So how many days? In 2 days, so I need to divide 9 by 2, so the final answer is 4.5. Right? Next question, that is the scientists concluded. So they told you what they can see, okay? That the soil mixture experiment showed there were different communities of bacteria in soil A and B. What is the evidence? So you, looking at this figure 6, are allowed to say nothing else than just a statement that says, yes, this conclusion is correct. What can I see from the figure 6 that tells me exactly the same thing as this conclusion? So what we can see, that at the pH 4.3, so sample B has faster rate of breakdown of ammonia. So you can see that from the graph. Next, A and B, okay, it's the slowest, we can see, and 4.3 doesn't work well at the pH 6.9, so the mixture. So this is nothing else than just looking at this and providing the reason for that conclusion that they stated. Right, so that's everything what they wanted from you, and almost there. So obviously another information here about the oxidation. We know what's that, and we've got the specific gene here coding for the production of ammonia monooxidase, and we've got more information here about the samples S and T. So the scientists grew those cultures separately in the soils at different pHs, and they determined the amount of mRNA from the gene. So copies of mRNA, this is what they're showing you, soil pH, and the species S and T at those different pHs. What can we clearly see here? Okay, so the species S, okay, look, so we've got the decrease here from 10 to power 6 to 10 to power 4, okay, then we've got species B, increase, so from 10 to 2 to 10 to 4, okay. So what was the question then? They're asking you in which species the number of copies are more affected, okay, by the changes in the pH from 4.9 to 7.5, and use the calculations to support this answer. So what's the calculation? From 10 times to power 6, take away that, and then this, take away that. So, so from there to there, and then from here to there, right, because that's the pH that I'm interested in. So once you've done that, we will see that species has changed by 990 thousandths per gram, but the other one changes by 9,900 per gram. Okay, so this is by looking at that standard form from the graph. So this method allowed the scientists to estimate the expression of the gene in each culture, but not the growth, and give the reason why. Why would you not be able to look at the growth? Because they didn't actually count how many bacteria was present in there. We've got no information. And number of mRNA, it's related to the amount of the enzyme only, okay? And we don't know how much mRNA is produced by each of the cells. We also don't know if actually this gene is linked to cell division, so we cannot estimate the growth based on that gene because it might not be its function. Right, and here we've got the scientists set up the cultures in the sterile glass bottles. Suggest to again give the reason for the method, so how for sterilizing the bottles, and explain why it's necessary. Okay, so method, what's the method? For example, washing the alcohol. Okay, and why would you do so? To remove any other bacteria that might break down ammonia, because this is what the investigation was about. We were looking at the ammonia. Right, so that's the end of section A for paper 3. Make sure you made a note of all of those exam tips. Look at the wording, look at the comment word. The fact is, if you know your stuff, so if you know your content, and if you fully understand the comment words, you can do it, okay? So please make sure you highlight the things, you write around the questions as I did, so make it visible, make it easier. Right, so that's everything for paper 3. See you later.